So today we're going to be talking about the transport of CO2 in blood. We've already talked about the transport of oxygen, specifically how most of the oxygen in the blood is transported bound to hemoglobin, and only a small amount that's dissolved. However, it's the amount that's dissolved, the PO2 of blood, that determines how much oxygen, as you well know, is bound to hemoglobin. And we covered that in detail when we looked at the oxygen-hemoglobin dissociation curves. So what we're doing today is, again, looking at how CO2 is carried in blood, not only in a dissolved form, but also in the form of, in a way it's bound to hemoglobin, and also in the form of bicarbonate. And again, these latter two involve a number of reactions that take place at the level of the red cell. We're going to use this figure here, and I'm going to reproduce this on the board slowly, and we'll come back to it as a summary. So if these are tissue cells, this is the interstitial fluid, and here we have a capillary, and in the capillary we have obviously plasma in a red cell. Now, the tissue cells produce CO2, which goes into solution in the interstitial fluid and equilibrates with plasma. So CO2 goes into solution in plasma. As CO2 goes into solution in plasma, CO2 will also enter the red blood cell and go into the solution in the red blood cell. So we have CO2 in solution. As the levels of CO2 go up, as the blood passes through this capillary, some of the CO2 that came into the cell and is in solution will react with hemoglobin. Specifically, the CO2 will bind the globin component of the molecule, not the heme, as occurs with oxygen. So it binds specific amino acids in the globin portion of the molecule. And we produce, as a result of this binding, we produce what is known as HbCO2, which is carbamino hemoglobin. Now, in the red cell, there's a very high concentration of a molecule, an enzyme, that is known as Ca that you've encountered before, and Ca is carbonic anhydrase. And carbonic anhydrase will catalyze a reaction between CO2 and water to form carbonic acid. So CO2 also reacts with water in the red cell. And due to the high presence of carbonic anhydrase, carbonic acid is formed, which then dissociates into hydrogen ions and bicarbonate ions. This is the source of the bicarbonate that basically is the predominant manner in which CO2 is carried in plasma. The bicarbonate is exchanged for a chloride molecule, and it leaves the red blood cell. So the chloride comes in from plasma. This is known as the chloride shift. So as blood passes through the tissues, the levels of bicarb in plasma go up, the levels of chloride in plasma go down. As a result of these reactions, about 60% of the CO2 that's carried in plasma is carried in the form of bicarbonate. Now, it's very hard to talk about CO2 transport in plasma without also talking about what's going on with oxygen. So with oxygen, as this blood passes through the systemic capillaries, the hemoglobin that had O2 bound to it, the oxygen dissociates. And the oxygen diffuses out of the red cell, out of the capillary, into the interstitial fluid, where it's going to be used by the cells. As oxygen dissociates from hemoglobin, we basically now have deoxyhemoglobin that can very quickly bind this hydrogen ion and therefore become reduced, producing this reduced form of hemoglobin. This is important later to explain how the transport of CO2 is intimately linked to the levels of oxygen in blood. Now, using the figure over here as a summary, we basically see the same series of reactions that we've talked about. We have CO2 produced by the tissue cells. CO2 basically gets dissolved in the interstitium and in plasma. It enters the red cell, as you see here in what's labeled as number 1. The dissolved CO2 binds hemoglobin, and now we form this carbamino hemoglobin. Some of the dissolved CO2 participates in the reactions with water in the presence of carbonic anhydrase, ultimately to produce hydrogen ions and bicarbonate. The bicarbonate is exchanged for chloride in the so-called chloride shift. Again, as this is going on, oxygen dissociates from hemoglobin, and the deoxyhemoglobin binds the hydrogen ions produced by this reaction to form a reduced form of hemoglobin. This is what's going on in the tissues. As blood passes through the pulmonary circulation, this is the second part of this diagram, basically these reactions occur in reverse. And CO2 ultimately diffuses into the alveoli, and ultimately the levels of bicarbonate in plasma are reduced. To outline what's going on, we basically see that because of the PCO2 gradients, dissolved CO2 comes out of solution and enters the alveoli. So there's a gradient moving, partial pressure gradient, moving CO2 out of solution into the alveoli. As a result of this, some of the CO2 that's bound to hemoglobin in the form of carbamino hemoglobin dissociates from hemoglobin. And this reaction goes in this direction. Likewise, the bicarbonate comes into the cell in exchange for a chloride, so a reverse chloride shift, and the bicarb combines with hydrogen ions that are released as hemoglobin picks up O2. And these hydrogen ions combining with the bicarb form carbonic acid, which then will break up into CO2 and water, and again the CO2 goes out of solution into the alveoli. So it's a series of reactions, just it's the opposite of what you see at the level of the tissue cells. Now there's a couple of points that are important to point out, again related to the relationship between O2 and CO2 transport. And the first thing is that as blood passes through the tissues, the hydrogen ions that are produced by this reaction, by the transport of CO2 and its conversion to bicarb, these hydrogen ions favor the dissociation of oxygen from hemoglobin. This is what is known as the Bohr effect. That is, increased hydrogen ions, either produced by the transport of CO2 and its conversion to bicarb, or, for example, produced in the tissues because of the generation of lactic acid. These increased hydrogen ions encourage the dissociation of oxygen from hemoglobin and allow hemoglobin to give up O2 more readily to tissue cells. This is one of the reasons why during exercise, even low to moderate intense exercise, there's always a baseline anaerobic metabolic activity that generates a small amount of lactic acid. And that's beneficial because the small amount of lactic acid that's generated at the level of skeletal muscle will encourage the hemoglobin to give up O2 as blood passes through those capillary beds. This is a phenomenon that is known as the Bohr effect. The other important concept is that the amount of CO2 transported in the blood is directly affected by the oxygenation of the blood. So in other words, the summary is that deoxygenated blood carries CO2 better than oxygenated blood. And you can clearly see that by looking at these reactions, which may be easier to see on the board here. As blood passes through the tissue cells, the hemoglobin gives up O2. As hemoglobin gives up O2, it very easily becomes reduced. And the source of these hydrogen ions is the carbonic anhydrase reaction. So by picking up these hydrogen ions and getting reduced, hemoglobin is pulling this reaction to the right, therefore encouraging the conversion of CO2 to bicarbonate. By pushing this reaction to the right and encouraging the formation of bicarbonate, the hemoglobin is basically increasing the ability of the blood to transport CO2. Likewise, the other important concept is that once reduced, this reduced hemoglobin can physically bind CO2 better than the non-reduced version. And as a result, again, we enhance the ability of this blood to carry CO2. So again, in summary, as hemoglobin gives up oxygen, it becomes reduced, pulling this reaction to the right, enhancing the formation of bicarb, enhancing the ability of blood to carry CO2 in the form of bicarbonate. The reduced version of the molecule can also bind CO2 better. This is what we refer to as the Haldane effect. So you both should be very familiar with these two concepts, which are summarized in this next slide. The fact that as CO2 binds hemoglobin, the affinity that hemoglobin has for oxygen goes down, but more importantly, that hydrogen ions generated from the transport of CO2 or from lactic acid reduce the ability of hemoglobin to bind O2. So as the levels of CO2 in blood increase, hemoglobin more readily gives up oxygen. And we talked about this when we talked about the oxygen hemoglobin dissociation curve. We just talked about it in a different concept. The Haldane effect is what we just covered a second ago. The simple fact that hemoglobin, as it becomes dissociated, enhances the ability of the blood to carry oxygen, simply because it siphons hydrogens from the carbonic anhydrase reaction, pulling the reaction to the right, and also because the reduced version of hemoglobin can bind CO2 better. Again, that's the Haldane effect. Moving on, we're going to talk about the significance of transporting CO2 in blood predominantly as bicarb. And the significance of that is that this form of transport of CO2 as bicarb creates what we call an alkaline reserve in blood that can be used as a protection against metabolic stresses that are acidotic in nature. So I'm going to put the carbonic anhydrase reaction on the board to the right here in a larger form than what we have there. And we're going to address the significance of the alkaline reserve. which is the bicarbonate we carry in blood. And first of all, the significance of the alkaline reserve in terms of protection against acidotic stresses that are metabolic in nature. A perfect example of this would be what happens as a compensation for diabetic ketoacidosis. Now, but any other metabolic stress that would result in increased production of acid would also be an adequate example. But let's use diabetic ketoacidosis as an example of such a metabolic stress that creates an acidic environment in the interstitial fluid and therefore in plasma. The reason this happens in diabetic ketoacidosis is because of decreased usage of insulin. Blood sugar goes way up, and a series of metabolic changes occur that ultimately result in the production of ketone bodies. The body basically is in a state of starvation and generates these ketones. The ketone bodies are acidic and dissociate in solution, producing increased levels of hydrogen ions, which lowers the pH. And can severely affect cell function. So what's going on is that the levels of hydrogen ions are going up. So as the levels of hydrogen ions are going up, what now happens is we begin to push this carbonic anhydrase reaction that occurs in the red cells. We begin to push this to the right. And we begin to produce increased levels of carbonic acid, which then breaks up to form CO2 and water. So in this type of situation, as a result of pushing this reaction to the left, we basically see an elevation of CO2, which, as you will see in a little, is a major stimulus for ventilation, breathing. So an individual going through this is basically going to compensate for the acidosis through the use of the respiratory system. In other words, we're going to take this extra acid that is produced due to metabolic reasons, in this case, the ketoacidosis, and we're going to combine the acid with the bicarb, generate carbonic acid, produce CO2 and water, and then you're going to blow out the CO2 because the CO2 stimulates ventilation. So for all practical purposes, you're blowing out the acid. You're undergoing what is known as a respiratory compensation for metabolic acidosis. This is not going to solve the problem. Sooner or later, this individual will need to use insulin to fix things. But this is basically reducing the levels of hydrogen ions to allow for adequate cellular function to continue. And the key thing is that you're using the respiratory system as a form of compensation. Sooner or later, the kidney will also kick in. And you're going to use the kidney to also get rid of some of these hydrogens. But again, that won't fix the problem. Ultimately, you need to use insulin to fix the problem. This simply buys time. But the point here is that by changing the levels of hydrogen ion in plasma for metabolic reasons can dramatically affect CO2 levels and ventilation rates. And that's what we have outlined as this first bullet here. By changing the levels of hydrogen ions, we can change the levels of CO2 in plasma and therefore affect ventilation and use ventilation as a form of compensation for this condition. The converse is also true, that changes in respiratory rate, as you can see over here, can have dramatic effects on the hydrogen ion concentration of plasma and therefore on blood pH. And let's outline that. So again, this reaction takes place in the red cell. And now again, we'll see how changes in respiratory rate can affect the hydrogen ion concentration in plasma. The example that we will use is a scenario where there's decreased ventilation, decreased breathing, maybe because of head trauma. Or you could also use the example of COPD. Okay? But let's do this because it's more dramatic. With a situation like this, ventilation is severely reduced, if not completely abolished. So as a result of that, we can't maintain the PO2 levels in the alveoli and the PCO2 levels that drive all of the other gas exchanges. So what you begin to see is you begin to see an elevation in the CO2 levels in arterial blood because the alveolar PCO2 levels are also elevated due to the lack of ventilation. The CO2 reacts with water in the red cell to form carbonic acid. And therefore now we increase the production of hydrogen ions and bicarb. So what we begin to see is we begin to see an elevation in the hydrogen ions in plasma due to a respiratory reason. This is a condition that is known as respiratory acidosis, as opposed to the previous condition where the source of the acid was metabolic in nature, and therefore that was known as metabolic acidosis. So here, the respiratory system is the cause for the acidosis due to impaired or lack of breathing. We're pushing this reaction to the right, and we're generating increased levels of hydrogen ion in plasma, which drops the pH of extracellular fluid and impairs cellular function. Because the causes of the acidosis are respiratory in nature, we can't use the respiratory system as a way to compensate. So therefore, the only way to compensate for this is to have the kidney try to get rid of some of these hydrogens, or to address the source of the problem and get this person breathing again. Because if we get this person breathing again, we reduce the levels of CO2. And by reducing the levels of CO2, we begin to push this reaction to the left. So the idea here is that the changes in respiratory rate can have very dramatic effects in the pH of blood, just as changes in hydrogen ion concentration can have dramatic effects on the CO2 levels and the ventilation rates. At this point, we've pretty much covered the ways in which CO2 is transported and the significance of the fact that it's transported in the form of bicarb, in other words, the alkaline reserve. And we'll wrap this discussion talking or outlining some abnormalities in arterial PO2 and some abnormalities in arterial PCO2. This graph over here shows you that at a normal alveolar ventilation, I should say, occurs when the PO2 is about 100 millimeters of mercury and the PCO2 is about 46 millimeters of mercury. So with those partial pressures for the two gases, you have normal alveolar ventilation of 4,200 mLs per minute. This is alveolar ventilation, not pulmonary ventilation. Now, any condition whereby, with hyperventilation, you basically will see an elevation of arterial PO2s because you're moving more air in and out of the lungs, and you're going to increase the PO2 in the alveoli, therefore increasing arterial PO2. And likewise, you will see a drop in the PCO2 of arterial blood because you're dropping the PCO2 in the alveoli. Now, on the other hand, with hypoventilation, so if you're in this range right here, you will see the opposite take place. With hypoventilation, as we previously discussed, you're not breathing as effectively. So as a result, you're not moving air in and out of your lungs. So the alveolar PO2s begin to drop. And because the alveolar PO2s drop, the arterial PO2s drop. And likewise, you'll see a rise in the partial pressure of CO2, both in the alveoli and in arterial blood. Now, what causes abnormalities in breathing? Specifically, what can bring about a scenario that's illustrated by this side of this graph? There's different possibilities. First of all, there can be a condition known as hypoxic hypoxia, where we have low arterial PO2s, and therefore we can't adequately saturate and carry oxygen by having it bind to hemoglobin, and this results from any respiratory malfunction. This is what you would see in COPD. Very common, anemic hypoxia, where you have a reduced oxygen carrying capacity of blood, either because of a decreased number of red blood cells, or else because there's not enough amount of hemoglobin, or if the hemoglobin is there but the structure is deficient. These are the two major causes of anemic hypoxia, either a physical decrease in the number of cells, or more common, some problem with the structure of hemoglobin, its ability to bind oxygen, or simply not having enough hemoglobin. Iron deficiency leads to a condition where there's not enough hemoglobin around, and therefore anemic hypoxia, and probably the major cause of anemic hypoxia, certainly at least in females. Carbon monoxide poisoning can also lead to this form of hypoxia, because it interferes with the oxygen carrying capacity of blood. These two are less common, these two forms of hypoxia. Circulatory hypoxia results from the inability to deliver blood to the tissues, either because of vascular spasm, arterioles collapse, and therefore blood can't get to the capillaries downstream. This happens in congestive heart failure for reasons that we've talked about, the inability of the left ventricle to adequately pump. And circulatory shock, when there's a massive dilation of arterioles throughout the body, and as a result of that, pressure drops and you can't adequately perfuse capillary beds. Anaphylactic shock would be an example of this. Last, we have histotoxic hypoxia, quite uncommon, but still a form of hypoxia. Oxygen is delivered to the tissues in normal fashion, but for some reason the tissues can't use oxygen. Often a cause of this would be poisoning and impairment, for example, interference or inhibition of the electron transport system. Cyanide poisoning impairs the ability of the electron transport system to do its job and results in histotoxic hypoxia. You simply can't use the oxygen that's delivered to the tissues. Finally here, An example of a couple of abnormalities in our arterial PCO2s. Any condition, hypercapnia is any condition where there's excess CO2 in blood. Major causes for hypercapnia, so this is what you'd see over here, are indeed hypoventilation. It's very commonly related to hypoxic hypoxia in the sense that if there's excess CO2 in arterial blood, there's probably decreased levels of oxygen in arterial blood because of, again, impaired breathing. COPD would be an example where individuals have hypercapnia, have excess CO2 in arterial blood, and naturally, if they don't undergo oxygen therapy, they would also have hypoxic hypoxia. Hypocapnia is the opposite, is you have below normal arterial PCO2 due to hyperventilation. These are terms that you should be familiar with, just as these, and just know what they refer to and how these forms of hypoxia or how these patterns of breathing can be caused. This is a table that I just included in the handout that is a mini glossary of terms that are used clinically to describe specific respiratory states. Some of these terms are the ones that we just covered, so this would be a good way to just, review these last two slides. At this point, we've finished a discussion of CO2 transport, and we're going to move on to the next handout, which deals with the regulation of breathing, or the regulation of ventilation. I will leave the carbonic anhydrase reaction on the board because an understanding of this reaction, again, will help us understand the regulation of breathing. Just keep in mind that this reaction occurs within the red cell. I'm going to remove this part here, but this occurs in the red cell simply because of the red blood cells' high concentration of carbonic anhydrase. The ventilation or breathing is regulated largely in an involuntary fashion by the activity of a number of neurons that are found in the pons and predominantly in the medulla. And collectively, these neurons are known as the respiratory control centers that are found in the brainstem. And again, remember, the function of the brainstem is involuntary in nature, and therefore most of the regulation of breathing is involuntary and automatic due to the functioning of the neurons that are found in these centers. You can voluntarily change breathing patterns because the cortex can affect the activity to some extent of these neurons. There are other inputs to these neurons, however, that are on a moment-to-moment basis more important in regulating patterns of breathing than the cortical inputs. And that's really the topic of our discussion here. Let's grab a drink. The major respiratory centers are the centers that are found in the medulla, that are shown in blue in this slide. And there's two groups, or actually three groups. There's the dorsal respiratory group, which is found posteriorly in the medulla. There's the ventral respiratory group that is found more anteriorly. And kind of above the two is a group of neurons that is known as the pre-Boettinger complex. And the pre-Boettinger complex, as you will see, is really the major regulator of breathing. Then we also have two centers in the pons. There's the pneumotaxic center and the apneustic center. And these centers are secondary in terms of setting the pace and the pattern of breathing, but they certainly communicate with the medullary respiratory centers. Now, your book, if you look closely at it, clearly indicates that the major respiratory center, basically the pace setter of breathing, is the ventral respiratory group. And that is actually not correct. If you look at the literature, it's clearly that the major pace setters of breathing are the dorsal respiratory group, because the dorsal respiratory group has connections with motor neurons that project to the muscles of inspiration, namely the diaphragm and the external intercostals. And it's the cyclical stimulation of these muscles that brings about breathing. The pace at which the dorsal respiratory group innervates the diaphragm and the external intercostals is set by the pre-Boatzinger complex. So the pre-Boatzinger complex is really the pace setter when it comes to breathing. And the way we regulate breathing is by regulating the rate of inspiration and relative to expiration. So again, your book is not correct by implying that the ventral respiratory group is the pace setter. It's the collective work of the pre-Boatzinger complex and the dorsal respiratory group. What I've done in the next slide is basically draw a schematic of these centers using our typical, cartoon figures for neurons. So over here, we have a neuronal network at the level of the dorsal respiratory group. And over here, we have the pontine centers, which are the pneumotaxic center and the apneustic center. On the right, we have the ventral regulatory group. Now notice that the dorsal regulatory group neurons have connections to motor neurons that project to the muscles of inspiration, which are the diaphragm and the external intercostals. Notice the date here. I drew this when I was in college, the first time I took physiology in 1986. And one thing that you see missing here is the pre-Boatzinger complex. And the reason it's missing is because at that time we didn't know that it was the major pace setter of the DRG. So to this figure you could add a loop over here which communicates with the DRG and which sets the pace of the DRG. And that would be the pre-Boatzinger complex. To make a long story short, the pre-Boatzinger complex basically is made up of neurons that spontaneously depolarize and produce action potentials. And when they spontaneously depolarize and produce action potentials, they activate this loop. So this loop begins to fire, and we stimulate motor neurons that project to the diaphragm and the external intercostals. The diaphragm and the external intercostals contract, and we inspire. The pre-Boatzinger complex, which would be over here, then ceases to fire. When it ceases to fire, the DRG no longer is active. And when the DRG no longer is active, we don't stimulate these motor neurons. If we don't stimulate these motor neurons, you don't stimulate these muscles. And therefore these muscles relax, bringing about expiration. So really it's the rate at which the pre-Boatzinger complex turns on and off sets the pace for the DRG. Now, the apneustic center and the pneumotaxic center certainly communicate with the DRG. We know that the apneustic center tends to be stimulatory in nature, and the pneumotaxic center tends to be inhibitory, either by inhibiting the apneustic center or directly by inhibiting the DRG. But we can eliminate the input from these centers and still have breathing occur. The pattern of breathing will change and it won't be as smooth, but breathing can still occur, therefore implying that the major pace setters of breathing are the DRG and over here the pre-Boatzinger complex. Now on the right you see the ventral regulatory group. And you see that the ventral regulatory group is active only during forced breathing, so forced inspiration and forced expiration, and it's still poorly understood how this would occur. But the ventral regulatory group all I want you to be familiar with, it has an inspiratory area and an expiratory area. The inspiratory area innervates muscles of forced inspiration. These would be accessory muscles of inspiration, like, for example, the scalenes or the sternocleidomastoid that help elevate the rib cage when they contract. The expiratory portion of the VRG, it goes to the accessory muscles of expiration, for example, innervating the abdominals and causing increased contraction of abdominals during expiration. So the idea is that when this is active, not only do we have innervation to the normal muscles of inspiration, but we also have innervation to the muscles of forced inspiration when you inspire, and to the muscles of forced expiration when you expire. The question is, how do the two communicate? That's still unclear. For the rest of our discussion, we're going to focus exclusively on this. So again, you can explain changes in respiratory rate by simply explaining or realizing that you can change the rate of breathing by affecting how often the DRG turns on and off. And that's done by the pre-Boatsinger complex. So the pre-Boatsinger complex, how many times per minute it spontaneously depolarizes and then shuts off, that determines the respiratory rate by explaining the rate at which the DRG turns on and off, and therefore the rate at which we innervate the muscles of inspiration. Now, we can explain changes in depth in the following way. But the greater the frequency of action potentials traveling through these neurons, the greater the amount of neurotransmitter released at these endings at the synapses with the motor neurons. So if I increase the frequency of action potentials traveling in these loops, I increase the stimulation of these motor neurons, and I increase the frequency of action potentials in these motor neurons, which enhances the release of neurotransmitter, namely ACH, onto these muscles, and therefore increases the stimulation of these muscles, and these muscles would therefore contract more forcefully. So increased frequency of stimulation, okay, of the dorsal respiratory group causes increased frequency of stimulation of these motor neurons, increased frequency of stimulation of the diaphragm and external intercostals, and increased force of contraction. Increased force of contraction means changes, namely an increase in the tidal volume. So you should be familiar with how you can use this circuitry to explain not only changes in respiratory rate, but also changes in depth, namely changes in the tidal volume. This can be summarized by this next figure. Here we have the DRG, and rather than showing it as a loop, I'm simplifying, and we're simply showing it to you as a big neuron. So these are the neurons that are driven by the pre-Boatsinger complex. They stimulate motor neurons that travel via nerves, namely the phrenic nerve, to the diaphragm and the external intercostals. And changes in the activity of these neurons will change the activity of these motor neurons and affect breathing, because it affects the pattern of contraction and relaxation of these muscles. Now, notice the top part of this diagram. There's many inputs onto the DRG, some that are excitatory, some that are inhibitory in nature. So there's inputs into the DRG that affect its activity, and therefore will affect breathing. Oops. These inputs are shown actually here. These are variables or factors that can affect ventilation by affecting DRG activity. So what we're looking at and what's outlined in this slide are these inputs that can affect DRG activity and therefore affect breathing. What are these inputs? Well, you will see that in the brain, specifically in the dorsal walls of the fourth ventricle, and the fourth ventricle is found in the medulla. So the fourth ventricle is found very close to the DRG. In the dorsal walls of this fourth ventricle, there's chemoreceptors. And these chemoreceptors will monitor the levels of hydrogen ions in the cerebral spinal fluid. There's also some of these chemoreceptors that are found in the extracellular fluid of the medulla. So not just in the walls of the fourth ventricle, but there's some of these chemoreceptors actually physically in the extracellular fluid of the medulla. And both of them, again, monitor hydrogen ion levels of either the cerebral spinal fluid or the brain extracellular fluid. To make a long story short, as you will see, elevations in the hydrogen ion in the cerebral spinal fluid or brain extracellular fluid will result in stimulation of these chemoreceptors, and these chemoreceptors will stimulate the DRG and increase ventilation. Also in the walls of large arteries, namely of the carotid artery, and you'll see exactly where, and in the aortic arch, there's also chemoreceptors that communicate with the DRG. Now these chemoreceptors monitor the PCO2 of blood, monitor the pH, and monitor the PO2 of blood. And in response to changes in these variables, they vary their stimulation pattern of the DRG and can affect, therefore, the pattern of breathing. I just want to correct something here, because elevation in the PCO2 of blood will stimulate the DRG. On the other hand, a drop in the pH and a drop in the PO2 of blood is a stimulus also for the DRG. So this is somewhat incorrect. There's also baroreceptors or stretch receptors in the lungs, and these stretch receptors are stimulated if the lungs are overinflated. And as a result, they will inhibit the DRG, preventing damage to the lungs. This is known as the Herring-Brewer reflex. There's also proprioreceptors in the joints that communicate with the DRG, and you will see that increased joint movement stimulates these proprioreceptors as an important stimulus also in DRG activity. Last but not least, as I said before, there's connections from the cortex to the DRG. So to some extent, I can regulate my pattern of breathing consciously by using the cortical inputs to the DRG. Now, when it comes to moment-to-moment regulation of breathing, the central chemoreceptors and the peripheral chemoreceptors are probably the most important inputs. So that's what we're going to be talking about, at least initially here. This is a diagram that shows you how the central chemoreceptors, which are found in the walls of the fourth ventricle, or found scattered in the medulla next to the DRG, in the brain extracellular fluid, how these central chemoreceptors affect the activity of the DRG. So the green here is the DRG. And it also explains how the peripheral chemoreceptors affect the activity of the DRG. The stimulus in question that is going to trigger the increased activity of these central chemoreceptors is an elevation in the PCO2 of blood. Now, the important thing to note is that there's a very high concentration of carbonic anhydrase in the brain extracellular fluid and in the brain cerebral spinal fluid. And the other important thing to note is that CO2 is completely lipid soluble. So it readily passes through the blood-brain barrier. So an elevation of arterial PCO2s means that the CO2 can easily get into the brain extracellular fluid and the brain cerebral spinal fluid. And we see an elevation in the PCO2 in the brain extracellular fluid. Now, because of the high concentration of carbonic anhydrase, this reaction takes place. And it's summarized in the slide. In other words, the CO2 is converted, it reacts with water, again because of the high concentration of carbonic anhydrase, and again this is in cerebral spinal fluid or brain extracellular fluid. We get production of carbonic acid, which dissociates into hydrogens and bicarbonate. So as a result of the high arterial PCO2s, we get increased production of hydrogen ions in the brain extracellular fluid, which is the major stimulus for the central chemoreceptors. The central chemoreceptors then stimulate the medullary respiratory centers, namely the DRG. We have increased ventilation. If we have increased ventilation, we blow off CO2, So we begin to decrease levels of CO2, which then shuts off this, or relieves this situation, and eventually shuts off the increase in ventilation. So the important thing to note is that the central chemoreceptors are stimulated not by the CO2 directly. The central chemoreceptors are stimulated by the hydrogen ions that are derived from that increased CO2. The other thing to note is that the elevation in the CO2 of arterial blood, so the elevated arterial PCO2, also stimulates, although weakly, the peripheral chemoreceptors, which then stimulate the DRG and complement the action of the central chemoreceptors. But complement is the key, because the central chemoreceptors are the major stimulators of the DRG in response to elevation of PCO2. But again, the central chemoreceptors don't respond directly to the CO2. They respond to the hydrogen ions produced by that increased CO2. The peripheral chemoreceptors, on the other hand, respond directly to the elevated PCO2 of blood. Something else to note is that this works fine, this system works fine if there's reasonable fluctuations or reasonable increases in the PCO2 of blood. And indeed, the PCO2 of blood is the major stimulus for ventilation acting through the central chemoreceptors. But in pathologies like COPD, as you well know, or other conditions where there's severe hypoventilation, because of the hypoventilation, alveolar and therefore arterial PCO2s can get quite high. Remember that normal alveolar, normal arterial PCO2s are 46 millimeters of mercury. If we get into this range, which would be common in an individual who has COPD, then this poses a problem, because the CO2 is elevated to the point that it can directly inhibit the DRG, and actually you could extend this arrow to the central chemoreceptors themselves. So even though this reaction is taking place and we're trying to stimulate the central chemoreceptors, if the CO2 levels are very, very high, there can be a direct inhibition of the respiratory center by this very, very elevated PCO2. And this is where the peripheral chemoreceptors come into the mix. As you will see, the major role of the peripheral chemoreceptors are basically emergency, life-saving roles, where PCO2 levels are so high that the medullary respiratory centers can become inhibited. And that is outlined over here, before we get into that. So, the relationship between the central and the peripheral chemoreceptors. Again, this is a summary of where they are located and what they respond to. Now, in order to see the relationship between the two, I'm going to give you an example of an experiment where we took animals, and probably rats, and we denervated the peripheral chemoreceptors. The peripheral chemoreceptors are very localized. They're found right in the carotid bodies, so of the carotid sinus, right there, and also in the aortic arch. So it's very easy to ablate these and to destroy these, which is exactly what was done in this experiment. So then, in these animals where the peripheral chemoreceptors were denervated, we basically did three things. We experimentally decreased the PO2 levels in these animals, while holding the other variables that are important, arterial PCO2s, and while holding the blood pH constant. Now, this in vivo would be very hard to do, but we're doing this simply in the lab, so I can change the PO2 values in these animals while holding everything else constant. So, if I denervate the peripheral chemoreceptors, and I begin to decrease PO2 levels in blood, what happens is that the ventilatory response that should occur, in other words, the increase in ventilation that should occur in response to the decrease in the PO2 is eliminated. And this basically indicates that the peripheral chemoreceptors are important in monitoring the PO2 levels of blood. So by having a situation where elimination or denervation of the peripheral chemoreceptors knocks out the ventilatory response that is produced by decreased PO2s is an indication that the peripheral chemoreceptors respond to the PO2 of blood. Likewise, if I increase arterial hydrogen ion levels while holding everything else constant, you don't see the increase in ventilation that you normally do. Again, indicating that the peripheral chemoreceptors are important in responding to changes in blood pH, specifically in this case to a drop in blood pH caused by increased hydrogen ions. So again, under normal conditions, increased hydrogen ions stimulate the peripheral chemoreceptors and you get increased ventilation. In these animals, because these are denervated, the increase in hydrogen ion is abolished. The increase in hydrogen ion abolishes the normal increase in ventilation. And then the third response that is interesting to point out is if we take these animals and increase arterial PCO2s while holding the two other variables constant, you see that the ventilatory response is reduced by the increase in ventilation, that is, that you see by elevation in PCO2 is reduced by 30%. So what this says is that the peripheral chemoreceptors do respond to some extent to changes in the PCO2 of blood, but only account for 30% of the ventilatory response that is produced by elevation in the PCO2. So the elevation in the PCO2 of blood causes an increase in ventilation. 70% of this increase is produced by the central chemoreceptors, 30% by the peripheral chemoreceptors responding to this condition. On the other hand, the peripheral chemoreceptors are the receptors that respond to changes in the arterial levels of oxygen and changes in the arterial hydrogen ion concentration. So again, what are the key roles of these peripheral chemoreceptors? And that's shown initially in this figure. So keep in mind that again, the moment-to-moment regulation of breathing is brought about by the action of the central chemoreceptors. However, the peripherals are important because they kick in and become critical in emergency life-saving situations, such as what's outlined in this slide. Here, we have a situation where arterial PO2s begin to drop. Now, if arterial PO2s drop from, let's say, 100 to 90 to 80, the peripheral chemoreceptors aren't stimulated. The peripheral chemoreceptors are only stimulated when arterial PO2s drop below 60 millimeters of mercury. And this is a scenario where, if you recall correctly, we begin to enter the steep area of the hemoglobin oxygen dissociation curve, which means that in this area of the curve, small drops in the PO2 of blood can drastically affect the ability of blood to carry oxygen. So when the arterial levels of PO2 get into this range, a couple of things begin to occur. Because of the low levels of O2, the DRG is inhibited, which impairs breathing. The low levels of O2 in blood also directly inhibit the central chemoreceptor function. So, generally speaking, if the arterial levels of arterial PO2 are in this range or below this range, the other thing that is going on is that the PCO2 is probably drastically elevated. And if you go back to this other slide, remember that a drastic elevation of PCO2 in this range directly inhibits also the medullary respiratory centers, the DRG, and the central chemoreceptors. So it's a double whammy. Not only are the decreased levels of oxygen inhibiting the respiratory center and the major drivers of breathing, which are the central chemoreceptors, but the elevated PCO2s that accompany this are doing the same thing. So now what happens is that in this situation, the peripheral chemoreceptors are stimulated. And now these begin to drive, the peripheral chemoreceptors begin to drive the medullary respiratory centers, increasing ventilation, increasing arterial PO2s, and probably, as you well know, decreasing arterial PCO2s. So in this situation, as the PO2 falls below 60 millimeters of mercury, the peripheral chemoreceptors become the major drivers of the DRG. and become critical as an emergency life-saving mechanism. This is another example of the emergency life-saving roles of the peripheral chemoreceptors. And in this example, we are dealing with what we discussed a little while ago, which is a scenario where hydrogen ion concentration in plasma is going up because of metabolic reasons. So the example of diabetic ketoacidosis. So we have an elevation of hydrogen ion not due to respiratory reasons, but due to metabolic reasons. So the hydrogen ion levels in plasma go up, and you may say, well, the central chemoreceptors are going to respond to that increased hydrogen ion levels because that's what stimulates them. Well, that's not the case, because hydrogen ions can't make it through the blood-brain barrier. And because hydrogen ions can't make it through the blood-brain barrier, they can't stimulate and have no effect on the central chemoreceptors. However, the peripheral chemoreceptors detect that elevation. And the peripheral chemoreceptors then increase and stimulate the activity of the DRG, which increases ventilation. If you increase ventilation, you're pushing this reaction, right, to the left. And you're producing more CO2, which now is a stimulus for the central chemoreceptors indirectly. And you blow off, because of the increased ventilation, you blow off this extra CO2. So again, you're getting rid of acid. That's due to metabolic reasons in a form by creating carbonic acid and by blowing it out and producing CO2 and water. So in other words, the peripheral chemoreceptors are important in the relief for acidosis. So generally speaking, again, moment-to-moment regulation of breathing is brought about by the central chemoreceptors and their indirect ability to monitor the levels of CO2 in blood by monitoring the levels of hydrogen ion in brain extracellular fluid. But the peripheral chemoreceptors, they basically are responsible for emergency life-saving responses in a situation where the PO2 of arterial blood dramatically goes down, or where the hydrogen ions and levels in arterial blood dramatically go up. This is a table that shows the influences of various chemical factors on respiration, looking again at decreased PO2 of blood, the effect on peripheral, the effect on central chemoreceptors, the effects of elevation of PCO2 on the peripherals and the central chemoreceptors, and the effects of elevation in the hydrogen ion concentration of arterial blood. So this is a very nice summary for everything that we have covered thus far. Now, to kind of apply some of this material and kind of get a better understanding of it, we're going to look at COPD and how the COPD is exacerbated by its ability to interfere with the regulation of ventilation. So keep in mind that if this is a capillary, this is the rest of the board represents brain extracellular fluid. And under normal conditions, right, we have CO2 in solution. We have in the red cell, so this represents the red cell in this capillary. And again, the outside of the capillary is brain extracellular fluid. We've got our carbonic anhydrase reaction. And then the bicarb gets kicked out in exchange for chloride, going back to the very beginning of the class. So under normal conditions, the CO2 is carried in solution. Some of the CO2 enters the cell, gets converted to bicarb. As we discussed earlier, some of the CO2 also is bound to hemoglobin to form carbaminohemoglobin. CO2 is lipid soluble, so even though these capillaries have have very, very tight junctions. CO2 can enter the brain extracellular fluid. And remember, in the brain extracellular fluid, we also have high concentrations of carbonic anhydrase. So again, the CO2 combines with water. If CO2 levels go up here, okay, then we produce more bicarb. If CO2 levels go up in plasma, more CO2 enters the brain extracellular fluid. We get more hydrogen ions produced, and these hydrogen ions are stimulate the central chemoreceptors, which then increase the activity of the DRG, and you blow off that extra CO2. So this is the summary of the moment-to-moment regulation of breathing. Now, in COPD, we have a totally different scenario going on here. And with COPD, you have a dramatic elevation in the CO2. So you may say, well, if there's a dramatic elevation in the CO2 levels in plasma, there's going to be a massive increase in the CO2 levels in the brain extracellular fluid. You're going to get a massive increase in hydrogen ions in brain extracellular fluid, and we're going to stimulate the central chemoreceptors a lot, which is really going to be driving their DRG to a big extent and really increasing ventilation. Well, that's not the case, and the reason being is the following. Because you're having dramatic increases, and by dramatic increases, I can 70 to 90 or even 100. The PCO2 can get into those ranges. Remember, normal PCO2 of arterial blood is 46, but in individuals who have COPD, it's not unheard to have them walk around with PCO2s in the 90s. So what this means is that you're generating a ton of bicarb, okay, by pushing this reaction to the right in the red cell. So the levels of bicarb go way up in plasma. Because there's so much bicarb usually doesn't make it across the blood-brain barrier. But because there's so much bicarb, it actually leaks through. So the levels of bicarb go way, way up. So even though you're making a lot of hydrogen ions that should stimulate the central chemoreceptors, because some of this bicarb leaks through, right through here. The bicarb that leaks through neutralizes these hydrogen ions, and therefore removes the stimulus for the central chemoreceptors. So what this means is that in individuals who have COPD, the central chemoreceptors and their ability to stimulate the DRG begin to peter out, because of the fact that all of this bicarb that is found in plasma leaks into the brain extracellular fluid and neutralizes the hydrogen ions that would be stimulating the central chemoreceptors. So what this means is that the drive to breathe begins to be reduced, and these individuals begin to breathe less deeply, and actually the rate of breathing begins to go down. If they begin to do that, then eventually what happens is the PCO2 of blood keeps going up, and the PO2 of blood keeps going down. And eventually, at some point, because you're removing the drive to breathe from the DRG, the drive to ventilate, the drive to stimulate the DRG, that is, eventually, at some point, the PO2 gets into the range of less than 60, the PO2 of arterial blood gets into the range of less than 60 millimeters of mercury. At this point, the peripheral chemoreceptors, if you recall correctly, right? At this point, the peripheral chemoreceptors become active, and they in turn now begin to stimulate the DRG, and the pattern of breathing gets altered, and they begin to breathe at a higher rate and with an increased depth. So the point is, if you look at an individual who has COPD, the peripheral chemoreceptors become the major drivers of the DRG, because of the fact that eventually, if they hypoventilate, eventually the PCO2 of blood gets below 60 millimeters of mercury. Now you begin to see the problem, because most individuals who have COPD are under oxygen therapy. So because of that reason, this is going on, and you're removing the drive of the DRG, and this process, as you saw, by buffering hydrogen ions in the brain extracellular fluid, will no longer stimulate the central chemoreceptors. So just as before, the central chemoreceptors in individuals who have COPD are no longer the drivers of breathing. In an individual who has oxygen therapy, however, this never happens. So because you are maintaining the levels of oxygen in plasma in a normal range through oxygen therapy, the hypoventilation that is caused by this never leads to a drop in the PO2 of blood in this range. And as a result, the oxygen therapy basically eliminates, the oxygen therapy eliminates the stimulus of the peripheral chemoreceptors to the DRG. So what's going to happen is sooner or later, the levels of CO2 are going to be so high that because of the inability to stimulate the central chemoreceptors and the direct inhibition of the DRG by these levels, the individual will stop breathing. And that's what eventually will kill an individual who has COPD. Especially, if they use high amounts of oxygen. So indirectly, oxygen therapy is contributing to the inability of these individuals to breathe. And you should be able to explain that. The reason we give them oxygen therapy is to maintain them comfortable, because when or if O2 levels get into this range, then they become very, very uncomfortable. So again, be able to explain why the DRG is important in the regulation of breathing, and what happens to the activity of the DRG in a situation of COPD, in a situation of COPD without oxygen therapy, and in a situation of COPD with oxygen therapy. At this point, we'll also move on and look at the effects of breathing, or how exercise brings about changes in the pattern of breathing. Just another application, just as we had the COPD application. And in order to do this, we'll start out by looking at this slide that you've seen before, which looks at the number of cardiovascular adaptations that occur during exercise. So the x-axis represents work, to the right, greater level of work, 0, no work. And then we're looking at a number of cardiovascular parameters that change, obviously, as we increase the work intensity. Obviously, stroke volumes go up. Heart rates go up. Stroke volumes also go up. The two contribute to the elevations in cardiac output that you're well familiar with. Pressure, mean arterial pressure goes up with exercise. The systolic increases more than the diastolic. And as you well know, that's because the total peripheral resistance is actually going down due to vasodilation of arterioles and muscle. Now the key thing to pay attention to, which is why I'm showing you this figure, is that obviously with exercise, oxygen consumption goes up. So in order to meet these demands, you've got to ventilate more. And indeed, ventilation goes up with exercise. And the question here is, what causes the increase in ventilation? Here we see 3 stages of ventilation changes that occur with exercise. Stage 1, stage 2, and stage 3. Stage 1 is shown right here, which occurs at the onset of exercise. And it's probably mostly cortical in nature. Just the thought of exercise on its own will cause an increase in ventilation, and that's mostly because the cortex is stimulating the DRG and preparing you for the exercise bout. So you definitely see a quick increase at the onset. Ventilation on the y-axis, minutes after onset of exercise on the x. Notice the ventilation keeps increasing, and you saw this in the exercise testing we did in lab. And that's known as stage 2, the slow, steady increase, a transient period of adjustment. And eventually, if you maintain the exercise intensity at the same level, you get a steady state, okay, where ventilation basically achieves kind of a very stable level. Obviously, this only happens, stage 3, only happens if you are working, if you stabilize the workload. So if the workload keeps going up, what would happen is stage 2 would continue to evolve. Here is an example where the workload is constant, so we have all 3 stages. In the lab, in the exercise testing, we basically had a progressive increase in the workload, and as a result, ventilation kept steadily increasing. So you have a stage 2 that just continues to go up. And that's shown in these series of figures. So in this figure over here, you basically have oxygen consumption on the x-axis, and oxygen consumption is a measure of just of workload, because again, oxygen consumption goes up as workload goes up. And then we're looking at what happens to a variety of physiological variables in response to this workload. And first, I want to call your attention to arterial levels of oxygen and arterial levels of CO2. So during this range of workloads, notice that arterial levels of oxygen and arterial levels of CO2, so oxygen here and CO2 there, are held very, very constant. Despite that fact, ventilation is steadily increasing. So what this means is that you can't say that the increase in ventilation that we see through here is due to an elevation of CO2 or a drop in oxygen levels of blood, because the O2 levels are very constant and the CO2 levels are very constant. So it's the increase in ventilation is not due to a change in blood gases. The question is, if that's the case, what increases the ventilation during this range? Well, probably the best explanation for this is that during that phase of exercise, what probably drives the increase in ventilation is increased stimulation of proprioreceptors in your joints that communicate with the DRG and enhance the activity of the DRG. It's possible that increases in body temperature can also increase DRG activity, and maybe even increases in epinephrine can result in this, but the major reason for this steady increase is the fact that the proprioreceptors in joints are probably being stimulated. Again, the blood gases, right here and right there, are very, very constant. And therefore, what I'm implying here is that since the blood gases are very normal, they can't be the stimulus for the increased ventilation. It's possible, certainly, that the cortex also does this, but this is the major reason for that ventilatory increase. Now notice that throughout this range of work, the lactate levels are pretty steady. Okay? But eventually, as you well know, you reach a specific exercise intensity where lactate levels shoot up. This exercise intensity and the heart rate at which this occurs is known, as you well know, as the anaerobic threshold. And notice what happens to ventilation at that point. It dramatically goes up. And the reason why ventilation dramatically goes up is because you're producing increased levels of hydrogen ion in the form of lactic acid. And the hydrogen ions basically cause this reaction that occurs in the red blood cells to progress towards the left, generating more CO2, and it's this increase in CO2 now that really drives ventilation. And notice that due to this dramatic increase in ventilation, the arterial levels of blood actually go up and arterial levels of CO2 actually go down because of the tremendous amount of air movement that occurs in and out of the alveoli. So again, steady increase in ventilation produced by the proprioreceptors, stimulation of the proprioreceptors in the joints. Once you get to the lactate threshold, however, the major reason for this big increase in ventilation is the elimination of acid, okay, that is being generated by lactic acid production, and the elimination of acid and the increased production of CO2. in plasma, which really stimulates the ventilatory response. This is a slide that looks at how various variables that are oxygen and carbon dioxide related change during exercise. And again, it's a good tool to use with this figure. That is it.